Hello friends, welcome to Bosons Botany, and in this video the topic is photorespiration or the process of photorespiration. In the previous video I have explained the theory part of this topic. Now I will take up the process. So as you can see, there are three main blocks here: chloroplast, peroxisome, mitochondria. So these three organelles are involved in the process of photorespiration. So oxygen entering into the first organelle, which is chloroplast, from atmosphere combines with RUBP. RUBP is acting as oxygen is here because oxygen is combining with RUBP. This will produce phosphoglyceric acid. Phosphoglyceric acid will convert into phosphoglycolate, which is a 2-carbon compound. This will release one molecule of inorganic phosphate to convert into glycolate. Glycolate will convert into glyoxylate and will move into the next organelle, which is peroxisome. Glyoxylate is again a 2-carbon compound. Glyoxylate is again a 2-carbon compound, and this will convert into glycine. But during this conversion, following two processes take place. First, conversion of glutamic acid into alpha ketoglutarate or alpha ketoglutaric acid, and the second step is formation of water and half molecule of oxygen, and this reaction is catalyzed by glycolate oxidase, hydrogen peroxide, and catalase. So two main steps occur during the conversion process of glyoxylate into glycine. Glycine is an amino acid, and the two processes are glutamic acid converting into alpha ketoglutarate or alpha ketoglutaric acid, and formation of one molecule of water and half molecule of oxygen, which is catalyzed by glycolate oxidase, hydrogen peroxide, and catalase. This amino acid, that is glycine, shifts or gets transported into the third organelle, which is mitochondria, and two molecules of glycine are produced, as you can see here. This glycine converts into the next amino acid, which is serine. But during the course of conversion, following steps occur: release of ammonia and release of carbon dioxide. So this step is very important because during photosynthesis we have seen the release of oxygen as the main product, but in photorespiration carbon dioxide is released during the conversion of glycine into serine, as well as ammonia is released, and this process is known as condensation. Also, one molecule of NADPH2+ is released. Here NADPH2+ is not produced, but it is released. The difference is very important. It is not formed or not generated as the assimilatory power, but it is released. So this step is very important, that is conversion of glycine into serine, where one molecule of carbon dioxide, ammonia, and NADPH2+, these are released into the organelle mitochondria. Now serine, which is the second amino acid, shifts or transported into the second organelle, which is peroxisome. Now the cycle is reversing back or going back. So serine is moved into the second organelle, which is peroxisome, and it is converted into hydroxypyruvate. Hydroxypyruvate converts into glycerate. During the process, again one molecule of NADPH2+ is released but not produced. This glycerate will move into the first organelle, chloroplast. Now if I give you the points here, these are some very important brief points regarding this topic. So the first point is, in photosynthesis, RUBP binds with carbon dioxide to release oxygen and glucose. So in photosynthesis, RUBP will act as carboxylase but not oxygenase, because RUBP is binding with carbon dioxide to release oxygen and glucose. But in photorespiration, RUBP binds with oxygen at high temperature and at high oxygen concentration to release carbon dioxide. So RUBP is acting as oxygenase in photorespiration, and photorespiration occurs at high temperature and high oxygen concentration to release carbon dioxide. It is a harmful process. Why? Because one carbon atom in the form of carbon dioxide is released, which was fixed. This means that in the organelle mitochondria, during the conversion of glycine into serine, there is release of one carbon dioxide molecule. This carbon dioxide molecule was fixed. Fixed means converted into glucose molecule. But this fixed carbon atom is released in the form of carbon dioxide, which is not a good thing for a plant. The second reason for its harmful nature is no ATP and NADPH2 plus formation. So no energy formation at all during the process of photorespiration. So I think this topic is clear. This is a very short, small topic and very easy to understand. You can correlate this with the Kelvin cycle, where the process is just the reverse. That is, carbon dioxide will bind with RUBP instead of oxygen to produce glucose. But here oxygen is binding with RUBP, and 3 organelles are involved in the process. In which organelle what type of reaction is occurring is very important. Like in mitochondria, release of carbon dioxide, ammonia. These two steps or these two processes are very important occurring in the organelle mitochondria. So thank you for watching this video.